Roger, Captain, thank you. I know, we need to iterate this to all of our departments and make sure that it doesn't get lost over the off season. I'll tell my guys if you tell your guys.
Do you guys know if we're having intervention this morning?
You're not on SPL, Bob.
I'm not sure. What kind of intervention?
I think
Communication from the public.
What?
Questions from the public.
Oh.
An interaction, okay.
Interaction, whatever.
An
I word.
It's about the drinking.
Okay, I follow now. I'm not sure, Lisa, sorry. I will try to find out. I did hear a rumor of that yesterday, but then I didn't follow it up.
0'd my 6-8 wraps.
I don't know if they're planning to do it because nobody's told me, but originally it was going to be at 10 a.m. or something like that.
Okay, well good, we have some time to figure it out then. I'll once we get settled, I'll try and get Steve and see if he knows. Steve didn't have any kind of definite news. He thought Megan was going to tune in around 9 Central, but that's the only info he had, so he'll let us know.
Thank you.
Yeah. I was just going to needle you for that. Constitution? Is that the year the Constitution was written? I think so. Oh, we're young, I was around then. What? I can't hear your jokes. What'd you say? Ah, I should have known. I could have guessed. Yep. Bumping. Wait. Okay. It's sure.
Okay, stand by.
We're not having any readings, are we? I guess it's Herc reading.
All right, science, we are settled in. Whenever you're ready, we have a solid hour to the bottom. So whenever you want to talk science, we are ready to talk science.
Okay, give us a few minutes. Okay.
No worries, we got plenty of time.
Erin, do you know how far we are from shore and from San Clemente Island? I can't hear you. I don't think you're on.
Sorry, it would help if I talked to you. We are 26.5 kilometers southeast of San Clemente Island, and we are 83 kilometers off of San Diego, Point Loma Light.
Do you know those in miles?
I know that 83 kilometers is 44 nautical miles, and I can check the other one. Stand by.
So we're southeast of San Clemente Island.
So we're 14 nautical miles off of San Clemente.
To the south.
Southeast.
East.
Yeah, I'll zoom out and you can get a feel for where we are.
Yeah, this isn't the best map. I don't know.
All right, so 14 nautical miles, I took it off the pyramid headlight there on San Clemente.
Okay.
And then, yeah, 44 nautical miles off of San Diego and Point Loma Light, which is over here somewhere. We're in here. No, we're wedged right between. exciting things all around. It looked like were the ships still kind of just on the horizon when you came up?
We're further
Yeah.
south than this.
Yeah. Yup. Cool.
Yeah.
Okay, thank you, Erin.
Yep, no problem.
Okay, I think we can get started and welcome anyone who's listening during our descent. Welcome to the California Biomes program. We are exploring the Southern California borderland. This is our 7th dive, I believe. We have one more left. The primary objective is to study the animals and microbes associated with mineral-rich hard substrates. These would be phosphorites and ferromanganese crusts that form on the very rugged topography associated with this highly tectonically active region off of Los Angeles and San Diego and areas in between. Today we're diving at the San Clemente escarpment. We'll be making our deepest dive of the series. Our target depth is about 1,778 meters. And because we've had really good success finding phosphorites and sampling those and the animals and microbes on those, we're going to be targeting ferromanganese crust today with our deeper dive. We're hoping to find rock formations with these crusts that can be readily sampled. So we'll be starting at the base of a rather steep escarpment, the San Clemente escarpment. It's about just over 40 miles from shore off of San Diego, and it's, I forgot, 26 kilometers, I've forgotten in miles, southeast of San Clemente Island. And we'll be hopefully landing onto some sediments. We'll be able to take sediment cores where the Jensen lab will be looking at the microbes and their biopharmaceutical potential. And our lab will also be hopefully getting some cores and looking at the infauna and maybe even the microplastics accumulating down there. And then we will start climbing the slope. We expect to cover a couple of different rocky ridges on the way up. The slope might be some of our steepest terrain yet on this cruise. And we'll be looking for ferromanganese covered rocks, but also new kinds of invertebrates, corals and sponges in particular, and the Jensen lab will be looking at the associated microbes and their different metabolic capabilities and natural products. I will now let Kira tell us a little bit more about the geology of the area.
Yeah, we are in one of the deeper basins along the escarpments and many banks that are in the Southern California borderland. Good morning. If you Google Southern California borderland, you'll see this really interesting rugged terrain with lots of basins and topographic highs like ridges and banks and escarpments. And yeah, as Lisa was saying, this is going to be one of our deeper dives in this region, hoping that in parts where it's a little bit less sedimented, we can find ferromanganese crusts growing on these rocky outcrops and see some new biology in these deeper depth ranges. The San Clemente Island is near a fault zone, so we might hopefully have some more geologists logging in today who are interested in the earthquake potential and all of the complicated faulting in this region. And for those that don't know, faults are lines along which rock is slipping or converging or diverging. And so, yeah, we will hopefully see some new species, some better ferromanganese crust than we've gotten in other parts, and see some interesting outcrop features along this deeper basin.
Gabe, do you want to talk a little bit about what your lab does with the samples?
Sure. So like Lisa said, our lab is interested in the microbe and the microbe community, the metabolomic capacity of these microbes, specifically looking for the microbes that make certain types of chemicals that are useful for medicine. A lot of the medicines that we use today originate from microbes. Medicines that we use to treat bacterial infections, for example, most of them come from microbes that we've studied previously. So for us, this is a new frontier. We're looking at the deep sea sediments for a source of these microbes, and it's very exploratory. We're gonna look for and study these habitats for the first time, or close to being the first to do this. So it's pretty interesting that we get the chance to do it here, and so we'll do this with new technologies in DNA sequencing. We'll isolate DNA. We'll also isolate RNA and study it in the lab, and we'll also do more classical ways of studying these sediments by growing them in the lab. And not every microbe grows when you like bring it from the depths and put it in a media-rich agar plate like we usually do. That's why kind of we resort sometimes to the DNA method, but it's still very useful to obtain microbes growing in the lab. Then you could produce cultures and then study what are those microbes producing, maybe isolate some compounds from those cultures. So that's kind of like our goals from this project. Yeah, very exploratory, and we're really hopeful that we could study, or this will bring a lot of research knowledge. So that's kind of like where we're going for.
Thanks, Gabe. So back here in the science row, we've heard from Kira Mizell from USGS, from myself, Lisa Levin, from Scripps Institution of Oceanography, from Gabriel Castro, and we have a data logger, Devin Vlock. Would you like to speak a little bit about what you're doing up there?
Yeah, so I'm Devin, and I'm a first-year master's student in Lisa Levin's lab. And so I'm just now getting into deep sea biology. I've had a background in conservation and management, and I couldn't be more excited to be working under Lisa, the director for the Conservation for Marine Biodiversity and Conservation, former at Scripps Institution of Oceanography. So I'm hoping from this cruise we can get some useful data to classify and maybe catalog some of the biodiversity that we're seeing on the ocean depths here for some conservation and management implications.
Want to talk a little bit about the data logging process also, what you do?
Yeah, so I've got a computer screen right in front of me, and as we're going along on our dive and we're seeing any types of notes or observations that could be helpful, whether it's the biology or the geology or even debris and trash, I'm noting that. And whenever we input that into the computer, we get an associated CTD data. So we're getting the oxygen, we're getting temperature, we're getting depth, and we're also getting lat and long. So anytime I put any information in the computer, we can find out where that point was. So when we're going back and analyzing the data, we can find out where we put this in.
Yeah. Very cool.
I'm also in charge of taking pictures. So any of the nice pictures that you see online, hopefully that was one of mine.
Thanks, Devin.
Yeah. Yeah, since we have lots of time, we can continue to chat a little about the geology of the region. I was just reading up more on San Clemente Island, and Southern California geologist Mark Legg is in the science chat. He's been giving us lots of amazing geology nuggets, especially on the structural geology of the region. And he's letting us know that the San Clemente Fault was the first fault recognized in California, based on the steep escarpment on the northeast flank of San Clemente Island. And that was published in a paper by W.S.T. Smith in 1898. So it's a very historical region for studying faults. It's a very cool fact. Thank you so much, Mark, for that. So if you look on your map, if you've ever looked at the Channel Islands and you see San Clemente Island there, the San Clemente Fault, or the San Clemente Fracture Zone, is running on the northeast side. And if you follow the northeast line of the island straight down southeast, That's where the fracture zone continues, and that's where our San Clemente Canyon actually is another word that they use for it other than escarpment is forming, and where we're going to be diving today. So hopefully we'll also see some interesting geology related to this historical and interesting fault.
Thanks, Kira. This dive will be a little different from our other dives because we will be deeper. We should have more oxygen in the water. Most of this cruise, we've been hanging out in the oxygen minimum zone between maybe 1,100 and 500 or 600 meters. And but the deeper water masses that come from Antarctic bottom water and the more polar regions have more oxygen than that isolated water mass that we have been studying. So we're hoping to pick up different kinds of biology, microbiology and macro megabiology because of that. So this should be fun.
Yeah, I'm really looking forward to it.
You guys want to review the data in the dive points, the waypoints?
Yeah, let's do that. Thank you.
Okay. All right. So as Lisa said, we're in here, if you can see my mouse, between San Clemente and San Diego, Point Loma Light. We're not very far from where we dove yesterday. That was kind of at the edge of this red circle. It's about, 14 kilometers away, I believe. Let me check that. Yeah, 12 to 14 kilometers, not very far. So zooming in, before I turn on the background maps, we're going to be moving to the northeast for all these dives, for this dive. And get our layers on. All right, so that's our bathy. Again, red is shallow and blue is deep. We're starting quite deep. Like Lisa said, the deepest dive so far at 1778 meters to start. And then we'll be moving progressively up slope. Slopes are, looks like 0 to 45. So the red would be 45 degrees. The blue would be 0 or very low slope. So we'll be starting in little to no slope, hopefully in some sedimented area, and then hopefully progressing really quickly to some steeper slopes with more interesting terrain to look at. And we also have backscatter, and the white we're hoping would be more of a rocky area. So that's what we're going for. And there's this cool kind of bump that it's hard to see in 2D, but I believe it comes in around AN. It's just like this little extra hill there that hopefully will be pretty interesting. Maybe if I turn on the contours, we might get a feel for it. Yeah, so it's right in there. There's this extra little bump that we had in the old multi-beam data and we were kind of suspicious of, but then we went over it last night and it's real, so it'd be cool to check that out.
So excited.
Thanks, Aaron.
Yep.
Yep, thank you.
It's always fun when a cool feature on some older data turns out to be real.
Yeah, for sure.
During our meeting yesterday night, we as we were going through this dive, or the dive plan, we also saw two lines going over the steepness of the mountain, and we were wondering if that was like an artifact of the initial mapping, but we were far away still. We were waiting. We had kind of like the guess that this could be an artifact because of the shape of the lines. Do you recall that?
Yeah, let's ask Erin. Maybe if she puts the backscatter back up, we could see if those lines went away. Aaron, do we see any fault lines running through? I guess it would be more on the slope map. I don't know, but
Let's pan around. I kind of recall when the 3D map was up yesterday and you guys were looking at it, I remember seeing a very linear feature that also looked like an artifact to me. If I recall, it was close to the top of the slope. Does that sound right?
do you
I think
remember,
so.
Gabe,
I think it was going through the slope, crossing it.
crossing it?
Like crossing it.
More diagonal from bottom to top.
Oh, like in
Uh-huh.
that kind of azimuth.
I think so.
Don't, let me see.
I'm guessing that's probably disappeared. There's still some ridges, but
Yeah, let's check out the.
we did run over again last night and collect more data. So
Yeah.
Take a look. I really like working in 3D, so I get really frustrated with what I have available up here. But this line is definitely artifacts where you're matching two multi-beam lines.
Okay.
As are all of these, right? You can see the edges of the swath.
Uh-huh.
But anything going diagonal, if it's not, following the track line or directly perpendicular to track line, then there is always hope that it's a real feature.
Cool. Thank you.
Yep.
Sorry, Gabe, can't hear you for some reason. Thank you, Aaron.
No problem.
So Lisa, what are you looking for when you're choosing dive sites like these? I mean, the ocean's a pretty big place. What makes you look at a particular place and be like, I want to sample right there?
well, I consult with my sidekick over here, Kira Mizell, and ask her where we are most likely to find phosphorite rocks and ferromanganese rocks, because for this cruise, NOAA Office of Ocean Exploration and Research is funding us to study to characterize the biology of those mineral-rich substrates, both the microbiology and the macrobiology. So we are looking for steep areas that are going to be exposed to currents that might help these precipitates form. And in the case of phosphorites, I think low oxygen also makes more phosphorus available and contributes to the formation of phosphorites. Now, all these minerals form over very, very long periods of time, so we're not necessarily looking at modern precipitation and formation, right, Kira? We're looking at precipitates that probably formed over millions of years in some cases. But we do look for steep topography where there's high backscatter, so we know that the ground is hard as opposed to being completely covered with sediments and exposed to currents, and then in some cases low oxygen. What else would you like to add to that?
Yeah, totally.
Oh, and other people have collected rocks in some of these places
that's also
and provided an important clue.
Yeah.
Don't want to forget about that.
Yeah, if there's been previously published data that has rocks collected at a depth range or on a topography type that you're interested in, that's always really helpful information. Yeah, we know that ferromanganese crusts, they can be actively precipitating still, especially in regions where there's really low sedimentation and they can continue to grow on exposed rocky outcrops. So we're definitely looking for that feature in the bathymetry, like Lisa said, that really bright backscatter where there's really, the beam is bouncing back off of something hard instead of being attenuated in the sediment. And then these slope maps that the nav team has been really, and the mapping team has been really helpful making for us, showing where we have steep slopes and hoping that that's where you won't have as much sediment accumulation for the case of ferromanganese crusts. And then, yeah, for phosphorites, we know that they form a bit shallower, and they're part of a particular portion of a Miocene sedimentary complex. And so we can use some knowledge from previous studies of where we found these rocks and what kinds of geologic conditions form them to help choose these dive sites. It is definitely a fun task to try and interpret the maps and make a good educated guess of where we're going. We've always got our exploration hats on. We never know what we're going to find, but we use as much oceanographic and geographic and geologic data as we can to pick the sites of interest.
Picking our dive sites, we also, on any given day, have to think about what we're trying to do. So for example, today's dive has very steep slopes. We considered it yesterday, but we knew we had to put out a series of colonization experiments, and we didn't want them to roll down to the bottom of the escarpment. So we chose to dive at 40 Mile Bank, where there were some more gentle slopes and some more terrace-like features. And even there, it was difficult to position the experiments in a way that they wouldn't roll downhill. So today's dive is likely to be even steeper. We have a comment from Mark Legg that tells us about some past dives in the 1980s that had upward-looking sonar that went from 1,800 meters to 5 meters, somehow all at one time, so that there must be very steep slopes there. We'll see. We know that our scale has changed. So, for example, yesterday on our slope map, the highest slopes were about 30 degrees, and now they're about 45 degrees on this map for San Clemente Escarpment. So we might find some steep rocks.
Excuse me, Aaron.
Yep.
Could we zoom out and see the canyon again, like we were seeing it before?
Sure.
Thank you. Here.
Would you like another background map on?
I think this is fine.
Okay. So that really, that artifact really shows up in the slope there.
Uh-huh. Yeah. Like these. Okay. Yeah. Yeah.
Aaron's looking at us talking about the maps back here.
Oh, that's cool. You could talk on SPL.
I'm curious. Yeah, we're sort of seeing the way that